Hello? Okay, I apologize for the Google Meet. It's a horrible application. Can you all hear me now and see the screen? Hello?
Yes.
Okay, I promise you no interruptions will happen again. So this is the last question. We will finish this exam. Here, this question, they want a conclusion for this investigation. What is this graph showing you? The amount of carbohydrate in the diet on the x-axis and the exercise time. How will you make a conclusion? What happens to the time of exercise the more you eat carbohydrates? What do you think?
Increases.
Of course, it increases. It's very easy. Now, two scientists carefully selected athletes for the three groups in their study. This question is important. It was important that the data from the three groups were comparable. Describe two variables that the scientists should have considered when selecting athletes. When you are choosing human subjects, what should you put in consideration? This is also a very important question. Yes?
Same
I can't
age.
hear you. Same age. Very good. What else? Same age.
Same gender.
Same gender. Very good. Same health status. They are non-smokers, all of them. Very good. Okay. If you want to look at the mark scheme, but exactly any two from same sex, same age, same mass, same diet plan. They all eat the same thing. Same health status, sport discipline, medical history. They're all healthy. So your answer is very good. This is a good question. Starch is broken down into reducing sugars. Again, the food tests are on every exam. Describe the method you would use to test for the presence of reducing sugars. Now, what are reducing sugars? Which experiment here will you mention?
Benedict.
Benedict, of course. Thank you. Of course, don't forget that this is the only experiment that needs what?
A water bath.
Yes, needs a water bath. Very good. And the water bath should reach a temperature of at least?
70 to 80.
Yes. State the reagent used to test for the presence of starch.
Iodine solution.
Yes.
Iodine.
We finished the exam. This is a very easy exam, in my opinion. It contains all the classic criteria for any paper 6, but the graph was not very nice. I don't think you will do the graph perfectly, but if you follow the rules and you value out the numbers and you put the units, you'll get most of the grade. I'll send you this picture on the WhatsApp group so that you draw it yourself and you show me the picture. This question was very easy. Paper 2 question was very easy. The investigation question is very easy. If you are good at investigation questions and you follow the steps, the investigation question was very easy. And the table was not nice. So in my opinion, I would see mistakes in the table and in the graph because the table was not cut clear, like the table was not very clear. Otherwise, this is the most recent exam, November 2023. If you want me, I can send you a sample of this exam because I don't think you have it. Okay. I solved with you a recent exam, very good. I am now open to any questions. Anyone wants any question? In the paper 6 document, yes, Kadi, what is your question?
Doctor, can you remind me how to measure to measure the germination of seeds?
Germination of seeds? Okay, very easy. Germinate, we just opened at seeds. You're very lucky. Now, seeds, when they germinate, they need several, they need some stuff for them to germinate. What are the things that are essential for seeds to germinate? Water, oxygen, and warmth. So it is water, oxygen, and warmth. And the investigation is any typical investigation, but just put in mind that the seeds, they need a few days to germinate. For example, if you are going to test the effect of pH on the growth of germinating seeds, you will get, let's say, 3 Petri dishes that contain 3 seeds. These seeds are of the same type. And you will put in you will have to, of course, provide everything needed for the seeds to germinate. You will give them some water, you will give them oxygen, and you will give them a suitable temperature. So you will put the water in the Petri dish. Usually we put it on a wet cotton. So you will put wet cotton here with the same volume of water. You will provide all 3 criteria needed for them to germinate. The only thing you will change is that you will change the pH, because this is what they want in the question. I'm going to put in one Petri dish, for example, an acid, in another Petri dish, a base, and in a third Petri dish, it will be neutral because it will act as a control. And I can use, of course, more Petri dishes. And I will keep it for 3 days. And after 3 days, I'm going to come back to my Petri dishes and I'm going to count the number of seeds that actually grew. This is the number of seeds that germinated, the number of seeds that showed a baby plant. I will count them. That's it. Where is the problem?
If it asks for the effect of temperature, how to
I will
measure
put
the
them in
optimum temperature.
I'll put them in different ovens. Cannot put the Petri dish in a water bath. I will put it in a
Yes,
different
but
oven.
what
It's
I'm
called an
asking is
incubator.
how I'm going to I'm going to measure the dependent variable.
Also, the dependent variable doesn't change. The dependent variable will be the number of seeds that germinated after 5 days. So I'm going to get the same 3 Petri dishes. I will put on those Petri dishes the same number of seeds. I will give them the same amount of water, the same amount of oxygen, and what I will change is the temperature, because you're asking about the temperature. I'll put each Petri dish in a different oven with a different temperature. That's it. After 5 days, I'll get the Petri dishes out. Maybe here only 1 seed germinated. Here 2 seeds germinated. Here all the 3 seeds germinated. I will count the number of seeds that germinated after a fixed number of time.
Okay, thank you.
Okay. Any other question? Now I put for you this
Doctor,
document here. Yes, my dear.
when he asked me about the concentration, how to calculate the concentration?
The concentration, it's a crossover equation. Give me an equation, give me a question and I will tell you. For example, I'll give you an example. You cross multiply. So for example, they will give you a table and they tell you concentration of the salt. I don't know what's going on here. So look, for example, the volume of the solution and the concentration of the solution. And they will give you several values. For example, when the volume of the solution was 10, the concentration was 200. When the volume of the solution was 20, what is the concentration of the solution? So what will you do here? You will cross multiply. You will take 20 times 200 divided by 10 to get the missing value. This is how you get the concentration. They will give you a set of values and one of the values will be missing. So you will simply get, or you will do a cross multiply. Do you understand it?
Okay, thank you.
Okay.
Yes,
If
yes.
you are solving and you stopped at a question, send me the question and I'll help you with it. Now, please remember your calculations because they ask for a lot of calculations. And you have here in your document all the possible calculations that you are expected to know, all the formulas. It's on the last page. You'll find it in year 9 notes and you'll find it in the document here. The percentage change, the final minus initial over initial times 100. And the percentage, just the percentage, this is the part over the whole times 100. For example, what's percentage? The sum over the total, this is average. Percentage sum over total times 100. For example, you got a 40 out of a 70 on an exam. What's the percentage? 40 divided by 70 multiply by 100. This is the percentage, okay? Concentration of solutions, you cross multiply. Don't use this equation. This is too advanced. I told you, you will use it next year. You just cross multiply as I showed you. Surface area and volume. Well, in the last exam that we just solved, they asked you for the surface area. Surface area is 6 times the surface times the surface. For example, this is 4 and this is 4. 4 times 4, this is 1 surface area for one side. All this times 6. This is the surface area. Okay? See, 6 times 4 to the power of 2. The volume is the length times the width times the height. You should know this from math. And we said the average is the sum over the total. The magnification equation, we did it, we just did it. Graph, we plotted a graph. Bar graphs, of course, please don't forget that the bar graphs are not fused. It's only the histogram that are fused, and you can start a histogram from 0, or you can make a small dash to represent the numbers that are previous, like if you're starting with a big number, and then you can start with the number or the range that you have in your table. Okay? What other question? I don't want you to memorize the experiments, by the way. Every experiment you have here is for you to understand only. I never said memorize experiments, because you can get a completely different experiment. You can get one experiment from what you took, or you can get a completely different experiment. So experiments, understand. Don't memorize experiments. I'm going to revise the oxen for you, because I feel that some of you have issues with the oxen. Okay, and then I might let you go. Now, the auxin, this is a hormone that helps in plant growth and plant sensitivity, because it will make the plant bend. If I have a plant, let's say this is the shoot, and there is direct stimuli, the sun is coming straight from above, what will happen? The plant will grow straight. Why does it grow straight? Because the auxin is going to be produced at a certain area. Where is it produced? Who will remind me? Wake up.
The tip.
Yes, the auxin is always produced at the tip of the shoot or the root, and then it doesn't remain there. It will diffuse to different parts of the plant. Because the stimuli here is direct from above and it is not one-sided stimuli, the auxin in that case will be evenly or unevenly distributed.
Unevenly.
Evenly.
No, the stimuli is not one-sided stimuli. The stimuli is not from here. The stimuli is straight above, so there is equal distribution of auxin. Okay, Mustafa, is this Mustafa?
No.
Okay. Do you understand where you went wrong? When the stimuli is straight above, and it's not one-sided stimuli, then there will be equal distribution of auxin. The auxin will be equally distributed on all sides of the shoot. That's why the shoot will grow straight above. There will be equal cell elongation. Did you understand that?
Yes.
Okay, very good. Okay, tell me. Let's say I have a different example. I have the sun coming from the side, and I have here the shoot. The shoot will move like this towards the sun. Positive phototropism. Can you tell me why?
Because the stimuli, which is the sun,
Yeah.
the auxin will distribute to the side that's near the sun.
No, it will not distribute to the side that's near the sun. You did not study. Auxin hates the sun. It's going to actually be distributed on the shaded side, on the side away from the sun. Side A, for example, it will not be distributed on B, or less will be distributed on B. It's going to cause that side, the shaded side, to undergo cell elongation. It's going to grow longer, faster than side B. That's why it's going to bend towards the sun. And this is positive phototropism. Did you understand where you went wrong? You need to
Yeah,
only
okay.
memorize that part, that the auxin, in the case of the shoot, it hates the sun. Hates the sun, no sun. It loves the shaded side, and it will stimulate cell elongation. Good.
Good.
Now, in the case of the shoot also, this is a shoot and it is horizontal and it is underground. There is no sun. This is a shoot. What is the stimulus that it is going to be exposed there? There is no sun. The only stimulus will be gravity that is down here. Now, the auxin, where is it going to be produced?
At the tip.
At the tip also. It's always produced at the tip, and then it will diffuse. Now, the question is, how will it diffuse? On the side of gravity or away from gravity? This is the only part that you need to memorize. I will tell you in case you forgot, even though I don't expect you to forget something like this when you have tomorrow a mock exam. It's going to accumulate on the side towards gravity. So the auxin, remember please, in a shoot, it loves two things. It loves the shade and it loves gravity. Okay? I'm talking about a shoot because the root is a different scenario. It's going to accumulate towards the side, towards the gravity, and it's going to also stimulate cell elongation, and it will make it grow upwards because this is a shoot. It does not want to grow towards gravity. It wants to grow upwards. This is going to be negative geotropism. Are you with me? And these questions can come in paper 6 or 4. I hope no one has questions. If you do, ask me. Now, in the case of a root, the root again is placed horizontally. It is underground, so our stimulus here will be gravity only, and we expect the root to move downwards. Now, what happens? Again, the oxygen is produced at the tip, and then it will diffuse. And in that case, also, it will accumulate on the side towards gravity. But in that case, it will stimulate cell elongation or inhibit cell elongation.
Inhibit.
Exactly. And it's very logic. Side A, or this side towards gravity, will grow slowly, and side B will grow faster. So side B will bend downwards. And this is positive geotropism. Clear? So these questions can come in paper 6, they can come in paper 4. And when we are doing any investigation, we use a device that is rotating slowly. This is my plant. And it is put on a device that is rotating, going around slowly. Why?
Equally distribute gravity.
Yes, even distribution of the stimulus. Very good, or gravity. And it cancels out the effect of gravity or light. And we use it as a control. And they do ask you, this is called a clinostat. It's used as a control to cancel out the effect of gravity or to cancel out the effect of light. It is used as a control to compare the results. Okay. Anyone has any questions? Now, this was an exam that did not bring any question on plants, but that's not the case. They usually get a question on plants. Anyone has any questions? You want to ask me anything? These investigations, are they clear? You want to stop me? You
Doctor.
want to?
Can you revise the investigations of transpiration?
The transpiration, there is a single investigation, which is the potometer. Where is it? Yes, it's right there. The potometer here is a device in which you have a plant that is attached to a column of water. And this plant is exposed to a certain environmental condition. Let's say it is exposed to too much light, changes in light intensity. We will change the light intensity by changing the distance. Or it is exposed to wind. We will use a fan. Or it is exposed to humidity. We'll put it inside a plastic bag. And what do? We start the experiment with the air bubble at 1 at the beginning of the ruler. And then we will leave this plant some time to adapt to the new environmental condition. And after this time, the air bubble will move. And we will measure the distance traveled over time to know the rate of transpiration. The more distance it traveled, the more the transpiration. This is the potometer that is used with the transpiration, and we only change the environmental condition, the wind speed, the light intensity, the surrounding temperature. Any questions?
Doctor, if it's asking to investigate the rate of water uptake, is it talking about transpiration?
Yes. Water uptake is transpiration. Okay. Photosynthesis, they always test the presence of the starch at the end of the investigation. Water uptake is transpiration. What else? Any other investigation? I'm going to put everything in front of you if you want to ask anything. Again, I am not telling you to memorize those questions off by heart. No way. I'm just telling you to understand the investigation. And in any investigation, the first thing you ask yourself are the variables, the independent and the dependent. Here, let's go over the plant investigations quickly. Here, they're measuring the rate of photosynthesis by counting the number of bubbles. Of course, this is not accurate. Okay. Here, again, the number of bubbles. And the independent variable here will be the light intensity. And why did they put a water heater here, a beaker, to absorb the what?
The heat from the light.
Yes, very easy. I think you got used to them now. And this is an improved method. Here, they did not count the number of bubbles because it's very inaccurate, and they used a graduated cylinder. Okay, a syringe here to measure the volume of the gas. And here they measured the number of bubbles produced also when there is different temperature. Here they use different water temperature, and they monitor the temperature using a thermometer. Okay. This here talks about the food requirements in different conditions. If you're pregnant, if you're breastfeeding. When you're breastfeeding, you need the most energy. When you're young, you need the most proteins. This is so easy. This is actually year 9. The location of the vascular bundles. This experiment also is very easy. They will put this plant part. We explained it in plants in colored water, and then they will look at the underside of the stem, and they will see that the water is arranged, or the color is arranged in this direction, showing you the location of the xylem and the stem. The location of the xylem and the stem is arranged in a ring. And the xylem is towards the inside. You should have studied and know all this by now. And these wilting, of course, we want to prevent transpiration, so we will reduce the number of stomata, thick waxy cuticle, hairs. They can ask you to draw a leaf. I think you are very okay with chromatography because you also took it in chemistry. We will get plant pigments and we will put it on filter paper and it will separate. And the heavier it is, the more it will stay at the bottom. And the lighter it is, the more it will move along the filter paper. Very easy. Here again, you will cover the different leaves with Vaseline. You will measure the starting mass and the final mass and the percentage change in mass. And the more it loses water, the more there will be a greater percentage decrease in mass. Of course, the stomata is found mainly on the upper or the lower sides of the leaf.
Lower side.
Lower sides of the leaf. Okay, I don't want to waste your time. I know that you are studying the photometer. We just revised it. While you're studying, if you have any questions, send me the question on WhatsApp. Okay, please practice your graphs because I'm going to get you a complete paper 6. I'm not going to remove anything or change anything. So it will be a typical question, a typical paper. Investigation with graph with, so please, your investigation, I told you in the recent mark schemes, in the recent years, the answer was always in the way that we always do it. Independent variable, steps, constants. Okay? So, any questions before I go? Measure these numbers. I mean, memorize those numbers. Any questions before I go? I'm asking for the last time. Anyone has any question before I go? Hello?
No, thanks.
Okay. Good luck tomorrow. Bye.